The second half of chapter 8 is all about enzymes. So enzymes are proteins that are biological catalysts. A catalyst is something that speeds up a chemical reaction without being used up or altered in the process. Enzymes are a class of proteins, but not all proteins are enzymes. So the keratin in your hair and nails is a protein, but it's not an enzyme. Okay? So what enzymes do is they help speed up chemical reactions by lowering enzyme barriers, and we're going to talk about what that means. So here in this example, we have sucrose, which is table sugar, and the hydrolysis of sucrose is spontaneous, meaning it has a negative value for delta G. It releases free energy. Okay? But you have sucrose potentially sitting in your pantry at home, and it's stable. It's not automatically quickly breaking down into its monosaccharides, glucose and fructose. In order for that chemical reaction to happen quickly, it needs the help of an enzyme called sucrase. So enzymes typically end in ASE, okay? And they usually are the name of the thing they break down. So sucrase breaks down sucrose, lactase breaks down lactose, and so on and so forth. Hydrolysis of sucrose is spontaneous. It has a negative value for delta G. It releases free energy. But that reaction doesn't happen fast. It doesn't happen immediately. And the reason it doesn't happen right away is because there is a thermodynamic barrier called the activation energy that prevents that favorable reaction from happening right away. Now, one way to get over this barrier is to simply add energy to our starting molecule, and we can do that by adding heat. Heat's a good way to speed up a reaction if you're in the chemistry lab. However, in biological systems, that's a problem, right? The reason a fever is a problem is because if your temperature goes too high, it starts to denature or unfold important enzymes and other proteins in your body, so your cells won't work properly. So biological systems can't simply raise the temperature to get things to go faster. The way that organisms solve this problem is with the use of enzymes. What enzymes do is they lower the activation energy barrier. Write that down. Enzymes work by lowering the activation energy barrier. And they accomplish this in different ways. So the thermodynamic barrier, okay, the activation energy, you can think of as a hill, right? And imagine this is a ball that we want to roll down the other side of the hill. Well, we have to push the ball up to the top of the hill. We have to add energy in some form to get them to go. But whatever energy we expended to push them up to this maximally unstable state is going to be released as it rolls back down. So net, we have no change in energy in the system here. But when it rolls the rest of the way down, the rest of the energy is released and the reaction proceeds spontaneously. So to push the ball up to this peak, up to the top of this activation energy, we want to get our reactants in this unstable form. And there's different ways to accomplish that. Some enzymes work by perfectly lining up the substrate so they're most likely to react. Some enzymes work by lining up charged particles that make the molecule more unstable, or some physically stress the substrate by bending it into a different shape. But what they all have in common is they lower the activation energy barrier. Because the energy that we added in to push it up to this transition state is also released as we roll down the other side of the hill, the amount of free energy that's released by the system is the same with or without the enzyme. The only thing that's changed is this activation energy barrier or hill. Okay. So write this down. The amount of free energy released by the reaction is the same with or without the enzyme. In other words, the reaction is equally spontaneous. It has an identical value for delta G with or without the enzyme. So enzymes are very specific, and their specificity comes from their shape. Okay, so one enzyme works on just one substrate. The substrate is the molecule that's the reactant. Okay, and the part of the enzyme that interacts with the substrate is called the active site. So simply we can illustrate this as like a little Pac-Man guy. Okay, if this is sucrase and this is sucrose, okay, it exactly fits the active site. If we have sucrase and we try to break down lactose, which is also a disaccharide, and it's the same chemical reaction, hydrolysis would split the two monosaccharides apart. Because lactose is a different shape than sucrase, it can't fit into the active site, and the enzyme can't drive that particular chemical reaction. So that's a vocab word that you need to know. The active site is the part of the enzyme where the substrate fits in. When the substrate interacts with the active site, the enzyme actually conforms to it, like two hands grabbing each other in a handshake. They fit to match each other. And so when the protein folds around the substrate when it interacts with the active site, that's called an induced fit. So when the induced fit happens, the active site of the enzyme is going to catalyze whatever the chemical reaction is. But when the substrate gets converted into the products, it's no longer the substrate. It doesn't fit the active site anymore. And so the protein folds back into its original conformation and it releases the products. The substrate has been converted to the products, but the enzyme remains unchanged. And that same enzyme can act on new substrates and repeat the process as many times as there is substrate to act on. Anything that changes the shape of the protein, specifically changing the shape of the active site, is going to make the protein non-functional and it won't be able to catalyze its chemical reaction. So if we change the pH, we'll change the hydrogen ion concentration, which can disrupt hydrogen bonds and ionic bonds, and if it's acidic enough, even covalent bonds. And that's going to unfold secondary structure, tertiary structure, and even quaternary and primary structure. Temperature is going to break hydrogen bonds first, which will disrupt first secondary and then tertiary and quaternary structures. Organic solvents will allow those hydrophobic residues to pop to the outside. Salt can disrupt ionic bonds, okay? So for a particular enzyme, there's a particular set of conditions that are ideal. So for example, human enzymes work best at human body temperature, 37 degrees Celsius. Thermophilic bacteria that live in hot springs, and their enzymes work best close to 80 degrees Celsius. Their enzymes wouldn't even work at human body temperature. Within our own body, there are different enzymes that work at different pHs. So the enzymes in our stomach work best at a very acidic pH. But if you shift them to the small intestines, which are slightly basic, they won't work at all. Whereas different enzymes that work in our small intestines require a basic pH, and if you shift them to the stomach, they won't work either. Some enzymes need partners or cofactors that help them function. So cofactors include metal ions, and if you have an organic cofactor, that's called the coenzyme. So for example, alcohol dehydrogenase, which breaks down ethanol, requires a zinc ion to coordinate the ethanol molecule to hold it into the active site. If you have the protein part of the enzyme without its cofactor, it's called an apoprotein or an apoenzyme. And if its cofactor is present, it's the whole enzyme. It's a holoenzyme. One way that enzymes are regulated are through inhibitors, and there's two different kinds of inhibitors you need to know about. Competitive inhibitors compete with the substrate for the active site. So if the inhibitor is bound to the active site, the substrate can't bind and it can't catalyze the chemical reaction. Allosteric inhibitors bind the protein somewhere other than the active site, but when they bind away from the active site, it causes the protein to fold into a different shape so that it no longer catalyzes that particular chemical reaction. So some inhibitors permanently bind to the active site. These are called suicide inhibitors because they kill the enzyme, but they also prevent themselves from blocking any other enzyme activity. So another way that enzymes are regulated is through interacting with other subunits of the enzyme. This is called allosteric regulation. So for example, you can have an enzyme complex made of 4 subunits, and there can be positive regulation where an activator protein will stabilize the complex in a form that allows it to work, or an inhibitor binding will turn all of the enzymes in the complex off at the same time. So again, this is allosteric regulation because the activator or the inhibitor is binding somewhere other than the active site. You can also have cooperativity where if a substrate binds one subunit of the complex, it causes all the other subunits to fold into the active conformation. So this is also allosteric regulation because the substrate binding on this subunit changes the shape of all of the other subunits. So it's not binding the active site of these subunits, and so it's allosteric regulation. Many metabolic pathways are regulated by negative feedback regulation. So the end product of the pathway turns the whole system off. So for example, in the synthesis of the amino acid isoleucine, it acts as an inhibitor to the first enzyme in this process. So this regulates how much isoleucine is present in the cell. If you have a ton of isoleucine, there's no need to make more of it, and so the end product shuts the system off. One other way that enzymes are regulated are simply where they are in the cell. So for example, the enzymes that do the process of cellular respiration are predominantly located within the mitochondria. And so by localizing enzymes to different parts of the cell, you can regulate their activity as well.